Look at the combinations you can have for a genotype. You can have two of the exact same allele, in which case we call you homozygous. Okay, homo meaning the same. And so we can say you're homozygous dominant, okay, if you have both of the dominant alleles. We can say you're homozygous recessive if you have both of the recessive alleles. The other thing we can use, the other word or other situation is if you have two separate alleles, such as this, in which case you can probably figure out that we're going to call you heterozygous. And because it's a mix of the dominant and recessive, we just say heterozygous. We don't need to say heterozygous dominant or heterozygous recessive. Okay, where am I? Okay, the next thing we're going to look at is phenotype. So, I said before that we can observe genotype. Okay, we observe traits. Your genes determine what you're going to see. And so a phenotype is just describing what you can see. You have a dominant choice or a recessive choice. Okay, so your phenotype, let's say if you're a purple flower, that is the dominant phenotype. And the recessive phenotype is the white flower. Now, the cool thing what we can do, just before I get into that, just as a side note, you may be wondering how can, for example, homozygous dominant, how can both of these produce purple flowers? Okay, and flowers that are just as purple as each other. Okay, wouldn't having two copies make it better or more purple? Well, how you think about that is that think about it as a parent giving you an instruction. Okay, it might be clean your room, okay, or it could be study for 2 hours tonight. Okay, so your mum's yelling at you to do that. If you're a diligent child, such as this child over here, you are going to study for those 2 hours. It doesn't matter that your dad isn't telling you to do anything. In a similar way, or in a similar way, if you have two parents that are both yelling the exact same thing to you, they're both saying study for 2 hours, having two sets of the same instruction is not going to make you do that more somehow. Okay, the instruction, okay, if it's the same instruction from here and here, it doesn't matter if you're getting it once or twice. You are going to carry that instruction out properly. And it's the same in your cells. It doesn't matter if you get two copies of the same gene telling you to do the same thing, okay, as opposed to if you get one copy of that instruction. If it's a dominant instruction, your cell is going to carry that out faithfully. Okay. Next point we need to make is that we can infer genotype from phenotype. You cannot look into an organism's cells and look at its genes, not easily anyway, to say, oh, you've got two dominant alleles. But what we can do is look at its phenotype and make those predictions using something called Punnett squares. I'll just give you a quick orientation to a Punnett square. What we're doing is we're representing the mixing of two gametes of the two options of alleles from one organism, I'll call it the mother, and we're going to represent that breeding or crossing with one or of two alleles from the father. Okay? So what we do is we put the two allele options for each of the organisms in their respective boxes. So let's say your mother is homozygous dominant and your father is homozygous recessive. All we do here is drop down these alleles into these boxes. Okay, so we're representing one organism, two organisms, three organisms, four organisms. So the four possible combinations of gametes. In this case, it just so happens that all of them will be heterozygous. Okay, because they're all going to get a capital A, a dominant allele from their mother, and they're all going to get a recessive allele from their father. Okay, so one example of when that might be helpful. Let's say you have a purple flowering pea plant. Okay, it could be either homozygous dominant or heterozygous. Both of these will produce that phenotype. What could you do to be sure in terms of reproducing that plant? I guess you could try and get a genetic test done in some way, but we're not going to worry about that. So what could you do to be sure? You could deliberately breed it or do what's called a test cross. Sorry. So I need to just bring up a blank board here. So we're going to do a test cross. You know that your plant can be either homozygous or heterozygous. We don't know. All we can do is choose to breed it with something. So a test cross is when you deliberately breed an organism with, well, I won't tell you just yet which one you're going to breed it with. Let's say you have your purple plant up here. I'm going to write purple. You don't know whether it's, well, actually, you can tell that it has at least one dominant allele. We don't know what the second one is. So we could pair it with a homozygous dominant homozygous dominant organism. We could do that. Let's see what would happen. Okay, we're going to get capital A, homozygous dominant, some form of organism with at least one dominant allele. Okay, so remember, we can't see these inside the organism. All we would see is the phenotype. And in this case, all of these organisms are going to have the dominant phenotype. Okay, because they all have at least one copy of that dominant allele. Is that helpful to us? Not really, because if you're, let's say, it doesn't matter what the second allele is. I have not gained any more information about this mystery plant. Okay, if it was homozygous dominant, I would still see this. If it was heterozygous, I would still be seeing this. So that doesn't help me at all. What we need to do is pair it. No, I'll leave that there. What we need to do is pair it with a homozygous recessive organism. And this is what a test cross is. Okay, you deliberately pair it with an organism that has produced an only ever expressed recessive phenotype. So its parents express negative recessive phenotypes. It expresses recessive phenotypes, or the recessive phenotype. And so here we have, no, I will wipe this. Here we have two options, because our mystery plant could be either homozygous dominant or heterozygous dominant. Sorry, heterozygous. Let's see what happens. We're going to pair it with a homozygous recessive. If it is homozygous dominant, by the way, you always write the dominant allele first. That's just a convention or a rule that we always follow. So if you pair it with a homozygous recessive, you will get four dominant phenotypes. Okay, but if it is heterozygous, remember these are the only two options for having a purple plant. If it's heterozygous, what you will see is you will get two dominant and two recessive. So this will be your ratio, 2 dominant to, my ratio sign, to 2 recessive. Okay, in this case your dominant would be 2 purple to 2 white. So in this case, depending on what you get, okay, 4 dominant phenotypes or 2 dominant, 2 recessive, you can make a decision about what your original mystery plant was. Okay, so that's how we can infer genotype from phenotype. As an aside, you may be wondering, oh, but why can't you do a heterozygous pairing? Why can't you do this? Now, I guess there's no real hard and fast reason why you can't, but let's say we have this. Sorry, I did those two wrong. The only problem is that this one should be and A. Okay. The only problem is that this four dominant to 3 to 1, okay, of dominant to recessive, okay, this is a bit more difficult to distinguish between. Okay, so let's say you breed these two together on the right. Okay, remember these ratios, it does not mean that literally you're going to have 3 dominant phenotypes and then 1 recessive. Okay, you may have 4, 5, 6 of these dominant phenotypes before you ever see this recessive phenotype expressed. Okay, so these two will look very similar to each other until you get one of these. So it's not incorrect. Okay, you could tell that from having this pairing. It's just not as obvious as having a 4 dominant versus a 1-to-1 ratio. Okay, with this 1-to-1 ratio or 2-to-2 ratio of dominant to recessive, you're more likely to see that earlier. Whereas a 3-to-1 ratio, it may take you 4, 5, 6, 7, 8, 9, 10 offspring before you can actually tell. Okay, so that was just hitting home the point that we infer genotype from phenotype.